Going to get this thing started. Thank you guys for being here. This session is being recorded and the video will be uploaded to the A+ College Ready Science YouTube channel. I'll also send direct links out to your teachers by the end of the day. If you haven't already, please mute your microphone and turn off your camera. That just makes the system work better. There's a lot of people doing these conferences now, and the easier we can make it, it just makes it all work better for us. The PowerPoint and the FRQs I'm going to be using today, I'll also send those out to teachers. So if you want a copy, just shoot them an email. The next one of these webinars is going to be on April 14th. It'll also be at 2, and it's going to be looking at experimental design and statistics. So that's one that a lot of you might want to look at. Anyway, today we're going to talk about gene regulation. So let me get started. If you have any questions, just unmute and chime in and ask anytime. That's why we're here. So the thing that you got to realize is that every cell in a multicellular organism contains exactly the same DNA. But even though that's true, organisms have lots of different kinds of cells that look completely different from each other, and they have very different functions. The only way that can happen is through what we call selective gene expression. Even though all the DNA is in every cell, only certain genes are active in, say, a muscle cell, and a different set of genes is active in, say, a skin cell. The rest of the genes are silenced. They're turned off. They're not expressed. They're not used to make proteins. So that's the way cells become different kinds of cells, through selective gene expression. Scientists have worked for a long time to determine how these processes happen. And really to figure it out, they started in the most simple case, which are prokaryotes, bacteria. So we're going to start off by talking about how genes are regulated in bacteria and prokaryotes. And then we'll move toward the end of the discussion today into how it works in more complicated things like us. So bacteria mostly regulate their genes through what are called operons. There are maybe a few eukaryotic operons, but most of the time when we're talking about operons, we're talking about bacteria, prokaryotes. An operon is essentially a group of genes, a group of related genes, and the other parts of the DNA nearby that help to regulate the expression of those genes, when those genes are on, when they're off. So let me say that again. An operon is a group of related genes and the other parts of the DNA nearby, which help to regulate those genes, when they're on, when they're off, when they're expressed, when they're not. You might think of an operon as a functional unit of transcription and gene regulation. And we're going to talk about the different parts, these parts, in a minute, but I'm not going to read that to you. We're going to look at a picture to talk about it. And we're going to start off by talking about an example of an operon. And that operon is called the lac operon. Here the lac stands for lactase. Now you notice the ASE at the end. Hopefully you know that ASE on a word in science means it's an enzyme. So lactase is actually a group of proteins that act as enzymes which help to break down the sugar, lactose, common sugar in milk. So these genes, the structural genes, are the genes that actually code for the proteins that make up lactase. So these are the genes we're trying to regulate, the structural genes. And again, in this example, these are the genes that code for the proteins that make up lactase. Now, all these other pieces are regulatory pieces. So just what we call upstream of the structural genes, there's what's called a promoter and an operator. The promoter is the binding site for RNA polymerase. Now, if you were in our last session, we talked about RNA polymerase quite a bit. It's the main enzyme that carries out transcription. It's the enzyme that takes the DNA template and uses that DNA template to make RNA. For a gene to be on, for a gene to be expressed, it first has to be transcribed. The DNA has to be used to make RNA. So the promoter is the binding site on the DNA for RNA polymerase. It's sort of a spot that says, hey, the gene starts here. The operator is in between the promoter and the actual genes, and it serves as a binding site for a repressor protein. Think of that repressor as like a roadblock. The blue thing there is the repressor protein. If it's there, RNA polymerase can't make it from the promoter to the genes. So when a repressor is present, transcription is blocked. And when transcription is blocked, the gene's not expressed. Proteins can't be made. If there's not a repressor on the operon, then RNA polymerase will slide across. It will do transcription, which is what we see happening here. DNA uses a template to make mRNA. That mRNA will then move out to the ribosomes. And what we see on this arrow, this is translation. This is when the ribosomes use the mRNA as directions for building proteins. These proteins together act as the enzymes lactase. Now, somewhere else in the DNA, and we see that here, it may be a site nearby these other parts. It may be a site in another part of the bacterial chromosome, but there's a gene somewhere else called the regulator gene. Sometimes you'll hear them called regulatory genes. You might even hear them called repressor genes. These are the genes that code for the actual repressor protein. So these genes code for the repressor protein. So notice that this lac operon is called inducible. I want you to think about what the word inducible means. To induce something means to turn it on. And if you put IBLE on the end of any word, it essentially means capable of. So inducible means capable of being turned on. So these genes in most bacteria, these structural genes, the genes for lactase, are usually off. Because here's the thing, most bacteria aren't usually exposed to lactose. Well, if there's no lactose in your environment, it doesn't make sense to make lactase, an enzyme to break down something that's not there. So when there's no lactose present in the bacteria and in its environment, these genes are off. They're not expressed. The repressor protein is bound to the operator. It's blocking RNA polymerase, blocking transcription. What causes this operon to be induced, to be turned on, is the presence of lactose in the environment and in the bacteria. So when lactose is present, notice on the repressor, there's a binding site where lactose fits in, sort of like a key in a lock. And when it fits in, it causes that repressor protein to change shape. Once it changes shape, the repressor protein no longer binds to the operator. It lets go. Once it lets go, RNA polymerase can slide across, transcription happens, mRNA is made, that mRNA goes out to the ribosomes, it gets translated, and lactase gets made. Well, that lactase starts breaking down the lactose present in the cell and in the environment. Eventually, all that lactose is gone, including this piece and this piece. And when that lactose is gone, that repressor goes back to its active form. It rebinds to the operator, and now the operon is turned off again. So I want you to think about this setup of operon sort of like a negative feedback loop. Something is made when it's needed, and when it's no longer needed, the process is turned off. It's essentially a way to conserve resources. Make proteins when you need them, turn off the process of making them when you don't. But the lac operon, again, is an example of an inducible operon, a group of related structural genes and the other parts of the DNA nearby, which helps to control those genes, which helps to regulate them. And these are turned on in the presence of some object in the environment or in the cell. Let's talk about sort of the opposite of an inducible operon, and these are repressible operons. The example that you always hear about for a repressible operon is the trip operon. We'll talk more about that in a minute. But let's first of all talk about what repressible means. Again, capable of, and repressed essentially means being turned off. So here are the structural genes in this operon. Because this is a repressible operon, these genes are usually on. In most cases, they're being expressed. But when certain changes in the environment takes place, their expression can be turned off. Let's talk about the trip operon for a second and what it is. So tryptophan is an amino acid that bacteria have to have that they typically don't get from their environment. So they need this amino acid to make their proteins. They typically don't get it from their environment. So the trip operon, these genes code for enzymes which help to make tryptophan for the bacteria. So when they don't have it in their environment, they need to be making these enzymes because these enzymes will make tryptophan for them. So again, these genes, these structural genes are normally on, they're normally being expressed. But now let's say that this bacterial cell finds itself in an environment where there's a lot of tryptophan. So now there's tryptophan everywhere. Well, it doesn't make a lot of sense to make enzymes to make something that's already there. So what happens in the presence of tryptophan is that tryptophan will bind to repressors, which up until this point have just been floating around in an inactive state. But when tryptophan binds to them, the shape of the repressor changes. And it changes so that now it fits to this operator. And remember, once the repressor protein is bound to the operator, RNA polymerase is blocked, transcription can't happen. And if transcription can't happen, translation doesn't happen. So the making of these enzymes stops. And that's going to continue to be true until there's no longer any tryptophan present. Once all the tryptophan in the cell or in the environment is used up, this piece right here will also be used up, which means that the repressor protein will change shape once again. It'll go back to its inactive form. And since it's no longer bound to the operator, transcription happens. mRNA is made. It goes out to the ribosomes. It's used by those ribosomes to make the proteins, which then make tryptophan for the bacterial cell. So again, another example of an operon, usually on, capable of being turned off. That's a repressible protein. One more time, an operon is a group of related genes and the other parts of the DNA nearby which help to regulate the expression of those genes. The operon is the main way that bacterial cells go about regulating their gene expression. So let's talk about how the similar kinds of things happen in eukaryotes. There may be some operons present in eukaryotic cells, but they're not common. It's not something we're going to hear much about. We're going to talk about 4 or 5 different ways that eukaryotes can regulate the expression of their genes. One of them has to do with the structure of chromatin. Remember, chromatin is the DNA in its unwound active form. Just before cell division, whether we're talking mitosis or meiosis, that chromatin will wind up into very tight balls and essentially form chromosomes. But when it's unwound, that's when it can be used. So that's when it can be transcribed and translated. When it's unwound and working, we can recall chromatin euchromatin. This is DNA that's being expressed. But it turns out there's situations that don't involve cell division where the cell can cause certain genes to bunch up, to coil up into what's called heterochromatin. And when that chromatin is all coiled up in a tight ball, the enzymes that do transcription can't get to it. If transcription can't happen, translation doesn't happen. So by balling up the DNA, essentially genes are being turned off. This can happen in a couple different ways, but one of the main ways it happens is through what's called methylation, the attachment of CH3 groups, methyl groups, to the cytosines of the DNA of a particular gene. When those methyl groups are attached, sometimes they're called methyl tags, that causes the DNA in that area to tightly wind up around the histone proteins, the proteins that sort of act like spools for the DNA. And that particular gene in that area is wound so tight, transcription can't happen, translation can't happen. Typically, methylation is permanent. So when those genes are turned off in a particular cell, they stay off in that cell. They're never going to be turned back on. This is one of the main things that helps cells to differentiate. It turns off certain genes in one cell. It doesn't turn them off in other kinds of cells. So the two kinds of cells become different because they have different groups of genes that are being expressed in the two different cells. A good example of methylation in humans is the bar body. You guys, I hope, know that that males have X and Y sex chromosomes. Females have two X chromosomes. Turns out, though, that a normal, healthy human cell only has one functional X chromosome. It's not a problem in males. We only have one X to begin with. But in females, there are two X's. So one of them needs to be inactivated, turned off. And the way that happens is that one of the X chromosomes is highly methylated, causing the DNA to wind up in a really, really, really tight ball that you can actually see under a microscope. It's called a bar body. But that chromosome in that cell will never be active again. Turns out that which X chromosome is inactivated, which one's methylated is random from cell to cell in the same organism. So some of the cells in a female have X chromosomes that they got from their mother that are active. In other cells in the same person, the X chromosome they got from their father is active. Let's talk about another kind of tag which helps to regulate DNA in eukaryotes, and that's an acetyl group, a COCH3 group. These groups can get actually attached not to the DNA directly, but to the histones, the proteins that the DNA winds around. And when they're attached, this causes DNA to unwind. When it unwinds, now it can be transcribed, it can be translated, it can be expressed. So acetylation is a way to turn on genes. Unlike methylation, acetylation is reversible. So this allows cells to turn on genes when they need those proteins, and then those acetyl groups can be removed and turn off expression of those genes when they're no longer needed. This sort of works like the feedback loops we saw with operons in bacteria. This is reversible. Methylation, permanent. But they're both tags which help to regulate gene expression in eukaryotes. Here we can see methylation happening, and it causes the DNA to wind tightly up around the histone proteins. When DNA's acetylation happens, it causes the DNA to unwind, be expressed. So we get expression here, and we get no expression here. And here you can see how the DNA is unwound once it gets acetyl groups added to it. The genes in here can be transcribed and therefore translated, therefore being expressed. Now let's also talk a minute about epigenetics. This is a huge new field in genetics. Epi means essentially on top of genetics in this case. For a long time, we thought that the only thing that really determined your genes, your genetics, were the sequences of nucleotides, the A's, the T's, G's, D's. Here's someone with a mic on. If you guys would mute that for me, unless you have a question.
Sorry, I'm late, teacher.
Oh, that's okay. No problem. So what they found though is that typically what will happen in a zygote, or at least the early part of embryonic development, the acetyl groups and the methyl groups that were attached to the DNA that the individual inherited from their parents, those are removed. And essentially you start with a blank slate. But what we found, there's certain conditions where certain methyl groups and certain acetyl groups actually get inherited. And this can be due to like the lifestyle of your parents. For example, if they smoked, certain genes you may inherit that are methylated that aren't supposed to be methylated. And that means that those genes never become active in you because of the lifestyle of your parents. This also can go on with bad exercise habits, bad eating habits, all kinds of lifestyle choices can cause certain genes in the offspring of those individuals to be either methylated or acetylated when they're not supposed to be. That's kind of scary. But it also has to do with twins. You know, identical twins start off with exactly the same sequence of A's, T's, G's, and C's. And for their first few years of their life, you can't tell them apart. It's really, really hard to tell very young identical twins apart. But as they age, you can start telling them apart. Because what's happening as they get older, because of things they do in their life, different sets of genes get either methylated or acetylated. And even though their sequences of A's, T's, G's, and C's are the same, as they age, different groups of those genes are turned off or on in different places, causing them to start of lifestyle habits and lifestyle choices. Things like smoking, drug use, lack of exercise, bad diet. Those things affect which genes are actually being expressed. So methylation and acetylation, when they're actually inherited from parent to child, those are essentially the main mechanisms of epigenetics, regulating which genes are on and which genes are off in an individual. Let's talk about a couple more types of gene regulations in eukaryotes, and then I want to get to a couple of FRQs to end the day. So another way that genes are regulated in eukaryotes is what's called transcriptional control. So we already talked about RNA polymerase and how it's the enzyme that makes RNA from a DNA template. For it to do that, it first needs to attach to a promoter, an area in front of the gene that essentially says, start here. In humans and a lot of other eukaryotes, there's an extra step. The RNA polymerase can only attach with the help of these other proteins called transcription factors. These are really important. They're specific, they're coded for by specific individualized genes. And these the regulatory sequences of DNA near the genes interact with these transcription factors, and that can either enhance transcription, cause it to happen better, or it can block it. It can completely inhibit the gene expression. So the presence or absence of specific transcription factors can either enhance transcription. Turns out in eukaryotes, there are certain groups of genes that are influenced by the same transcription factors. So if these transcription factors are present, in a lot of cases, all of those related genes will be expressed at the same time. It's a way to coordinate the gene expression of multiple different genes. There's also what's called post-transcriptional control. So once pre-RNA is made, or what we call the primary transcript is made, remember what happens next is RNA processing. The introns are removed. The exons are spliced together. And this can happen in different ways, alternative processing. This allows one gene to actually make multiple proteins. The processes that control how that RNA gets processed, that's what we're talking about here, post-transcriptional control. Here we can see that happening. Here's one gene. made of exons, and in light blue, these are introns. Here's the primary transcript of RNA. Notice it contains both the introns and the exons. But then we get RNA processing and alternative splicing. This RNA ends up with all six exons and codes for this protein. But in another situation, when body chemistry is different, when things in the environment are different, exon 2 is essentially treated as an intron, and it gets removed. So we get a different mature mRNA, a different protein, and therefore a different expression of the gene, a different trait per se. Here, notice that we kept exon 4, but exon 2 is removed. That gives us a completely different protein. So this whole idea of one gene can code for multiple proteins depending on how it's processed. So by controlling the way it's processed, that's another way to control how genes are expressed. There's also a level called translational control. There are small pieces of RNA, sometimes they're called microRNAs, that can bind to and essentially block the translation of mRNAs. For example, in an ova, an egg cell, lots of genes that a developing zygote are going to need are transcribed. And those little RNAs, those mRNAs, are floating around all in this zygote's nucleus. But most of them have attached to them these little microRNAs to block translation. And I guess a good explanation for that is that most eggs are never fertilized. They're ovulated and they die. So it doesn't make sense for the body to use a lot of energy and a lot of effort to make proteins that are simply going to be wasted. So the microRNAs will stay attached to the mRNAs and block translation unless fertilization happens. When fertilization happens, these microRNAs are removed, and very quickly those mRNAs are used to make the proteins that the developing zygote will need. One final way that gene regulation in eukaryotes happens is through post-translational control. Remember, translation is the process of building polypeptide chains of amino acids. Most proteins are made of multiple polypeptides that wrap together in some three-dimensional shape. That wrapping and shaping happens in post-translational control. So by controlling how a protein folds, and also by controlling the lifespan of the protein, because there are enzymes that break down proteins, eukaryotes can essentially control gene regulation. Or gene expression, I should say. So those are some different ways that eukaryotes control expression of genes. Whereas in operons, or in bacteria rather, we're mostly going to talk about operons. I'm going to switch slides here for a minute and talk about an FRQ. So let me make this a little larger. And let's see if I can't make it scroll down. Just a minute to find the right question here. All right, here's the question I'm looking for. So again, let me make it a little larger. And let's see if I can't make it so you can read it. All right, looks like that's as big as it's going to let me make it. So I'll read it with you. A researcher is studying patterns of gene expression in mice. The researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of mRNA from 6 genes. The data are shown in figure 1. So here's figure 1. And notice we've got gene E, gene F, gene G, gene H, gene I, gene J. And in these different types of tissues, liver, heart, brain, kidney, pancreas, skeletal muscle, we have essentially a measure of how much mRNA there are from those genes. And notice if it's dark black, that means there's a lot of mRNA from that gene. If it's gray, there's some. And if it's white, none. So I want you to think about part A for just a minute. Part A says, based on the data provided, identify the gene that is most likely to encode a protein that is an essential component of glycolysis. Provide reasoning to support your identification. So one more time. Based on the data provided, identify the gene that is most likely to encode a protein that is an essential component of glycolysis. Provide reasoning to support your identification. So I want you to take about a minute or two and just mentally answer the question. You don't have to write anything down. Pay attention, though, to the verbs. Identify and provide reasoning. So take about a minute and a half. See if you can't answer this question in your mind. Just part A for right now. All right, so let's first of all think about what glycolysis is. Remember, glycolysis is the first step in cell respiration. It's the first step in both the aerobic and anaerobic versions of cell respiration. So without glycolysis, you're not going to make any ATP, and therefore your cell is not going to have any energy source. So I'm hoping that what you get from that is that every cell needs to have this protein, because without it, they don't have any energy. They can't access any energy. So if you look, what you want to think about, since every cell has to have it, this gene, the one that codes for this protein, should be the one that's present in every cell. And the only one that you see present in every cell is G. So essentially, you're going to identify, so you're going to say that gene G is most likely to encode the protein that's essential for glycolysis. And then your reasoning is going to be every cell has to do glycolysis in order to make ATP. Therefore, the mRNA from the gene for this protein must be present in every cell. And the only gene that has mRNA from it present in every cell is gene G. So you might answer the question in 2 or 3 sentences. I'm going to, I'll show you the rubric in a minute. Let's go ahead and look at part B as well. It says the researcher observed the tissues with a high level of gene H mRNA did not always have gene H protein. Provide reasoning to explain how tissues with high gene H mRNA can have no gene H protein. So even though the mRNA is present, there's no protein. If you think back to what we talked about a minute ago, so we see that gene H mRNA is present in the brain tissue, the kidney tissue, the pancreatic tissue, and the skeletal muscle tissue right in here. If you remember back just a second ago when we were talking about ways eukaryotes control gene expression, we talked about post-transcriptional levels of control. That even though the mRNA is made, there are ways to block the translation of those mRNAs. We talked about microRNAs and how that those microRNAs can bind to an mRNA and essentially prevent it, at least temporarily, from being translated. So I'm going to pull up the rubric for the scoring guidelines for this question real quick. Give you an idea of what the graders actually use to grade the question. So here's our question. And again, this is the way you could earn points on this question. So for part A, identify was gene G. And the reasoning was gene G is the only gene expressed in all 6 tissues, and glycolysis occurs in all 6 tissues. You had to say both of those. Or you could have said mRNA, gene G mRNA is the only mRNA present in all 6 tissues, and glycolysis has to occur in all 6 tissues. If we scroll down a bit, so for B, really only got 1 point here. You could have talked about sometimes mRNA can be, the transport of mRNA out of the nucleus can be blocked. We could say gene H mRNA is not translated. There's some kind of RNA interference going on. This would be the microRNAs attaching to it. Or you could have just talked about the idea of post-transcriptional control or post-transcriptional modification. All right, I'm going to look at one other FRQ for today. Let me get that one pulled up. Let's try that again. It's like I just lost my question. Give me just a second, guys. All right, so we're going to go to gene regulation FRQ2. And let me find the question we're looking for here. So we're looking for question 3. Make it a little bit larger. Let's see if we can't scroll down a bit. So question 3, information flow in cells can be regulated by various mechanisms. Describe the role of 3 of the following in the regulation of protein synthesis. All right, so let's do that. RNA splicing. Remember, RNA splicing is that idea of RNA processing. That's another way of saying it. So that depending on the situation going on in a cell, RNA can be processed or spliced different ways. This ends up leading to the translation of different proteins from the same gene. I think that's enough probably there, but we'll look at the grading rubric in a minute. Repressor proteins. So repressor proteins are proteins that can bind to the DNA and can block transcription and therefore essentially stop expression, stop protein synthesis for those genes. And then
brownies ready, man, to go.
Methylation is the attachment of CH3 groups directly to the DNA, causing it to wind up in essentially to a tight ball. And when it's in that form, the DNA can't be transcribed. And therefore, the genes that have been methylated are off. I doubt you've talked about siRNAs. So when we look at the grading rubric here in a second, we'll talk a little bit about those. But probably the three you guys should have picked on this question would have been RNA splicing, repressor proteins, and methylation. Let's go ahead and talk about B. Information flow can be altered by mutation. Describe 3 different types of mutations and their effects on protein synthesis. Tell you what, I'm going to go ahead and pull up the rubric on this one because I think I can get through this faster by looking at the grading rubric. All right, so here we go. Let's go ahead and look at part A. So describe the role of 3 of the following in the regulation of protein synthesis. We talked about RNA splicing. The idea that exons are spliced together, introns are removed. Spliceosomes help to do that. And that because they can be spliced in different ways, one gene can make multiple different proteins. We talked about all those things in that question. Repressor proteins. They can block transcription. They can block translation. Therefore, they silence genes. They inactivate the expression of genes. You could have said any one of any of these bulleted items for each item. Methylation. This is when methyl groups attach to DNA, prevent transcription. They also can protect it against restriction enzymes. That's probably not something you would have talked about here, though. I think most of the time you would talk about methylation causing the DNA to wind up and essentially be inactivated or inhibited. And then the siRNAs, those essentially are another way to talk about microRNAs, I guess. They facilitate the degradation of mRNAs. They can help it to be broken down. They also can block the translation of certain mRNAs. I want to scroll down and look at part B. So part B is not so much directly related to our topic for today, but this is a topic we talked about last time. Describe 3 different types of mutations and their effect on protein synthesis. So we've talked about silent mutations. Silent mutations are where you have a codon that, let's say it was AAA, and it coded for a certain amino acid. Well, then there's a mutation. And instead of AAA, we get a substitution where it becomes ACA. But it turns out that if AAA and ACA, if those codons code for the same amino acid, you still get the same amino acid sequence. You ultimately end up with the same protein, and it's like nothing happened. So essentially, silent mutation can be a change in a single nucleotide, a substitution. You don't get any change in the amino acid sequence. You don't get any change in the protein sequence. Therefore, there's essentially no effect. A missense mutation is a type of point mutation where there is a substitution. So a nucleotide is changed. Maybe we were supposed to have AAA, but instead we get ACA. And AAA and ACA code for 2 different amino acids. So we get a different amino acid, we end up with a different protein sequence, and that protein doesn't function the way it's supposed to function. A nonsense point mutation is similar. So you get a substitution again, but this time a codon that was supposed to code for an amino acid is now mutated, and it's a stop codon. So that the transcription stops in the middle, or translation stops in the middle, and the protein is truncated or it's never made. So it's essentially non-functional. You also could get frame shift mutations. So these are insertions or deletions where you have 1 or more nitrogenous bases that are inserted, and that throws off the whole reading order of the amino acid. So it essentially changes the amino acid sequence from that point forward for every amino acid. And this causes essentially a non-functional protein, or it can cause codons to become stop codons and truncate the protein very early in the process. You also could go into even things like duplications and inversions and non-disjunctions, which are essentially not gene problems, they're chromosome mutations. So we could have even talked about those. But I would guess in most cases, most of you probably would have talked about these first 2 or 3. And notice it does say 3. So if it specifically says to describe 3, only describe 3. Readers are only going to describe the first 3. Also pay attention to the word describe. It doesn't say identify. So you've got to say silent mutation and talk about what a silent mutation is and what effect it has. Say missense point mutation, talk about what it is and the effect it will have on the protein. You're not going to get any points for just saying silent, missense, and nonsense. You'll get 0 points for that. All right, scrolling on down to part C. Part C says identify 2 environmental factors that increase the mutation rate in an organism and discuss their effect on the genome of the organism. So things that can increase mutation rates, we call mutagens. Mutagens are environmental factors like UV light would be an example. Carcinogens, things like cigarette smoke, asbestos, radon gas, different forms of radiation like X-rays and gamma rays, and also certain kinds of chemicals like nitrites, even like Roundup you hear about on those lawyer commercials on TV. There are also viruses that can disrupt the gene sequence. Point being, what you need to talk about is that the DNA sequence is somehow changed. I think if you just said the DNA is altered or damaged on any of these, you would be okay. Essentially, thymine dimers, DNA is altered, damaged, disrupt gene sequence. Essentially, all these bullets on the discussion part are saying the DNA sequence has somehow changed. So identify 2 mutagens. So you might have chosen UV light and gamma rays. And then for each one, say that they cause the sequence of the DNA to be altered in some way. All right, final part of the question was D. And D says epigenetics is the study of heritable changes in the phenotype caused by mechanisms other than changes in the DNA sequence. Describe one example of epigenetic inheritance. So what we talked about with epigenetics were methyl groups or acetyl groups, which were actually inherited by a child from their parents. And in this list that you can see here, the example is a DNA or histone modification. Methyl groups modify the DNA, acetyl groups modify the histones. So that's what I would have talked about. We also talked about bar bodies being an example of methylation. Heterochromatin is after DNA has been methylated and it winds up. Some of these others we didn't discuss, but they're things that could be inherited. Like, for example, maternal diet can affect which genes are on and which are off in an offspring. Heavy metal poisoning. Here we're talking about like mercury, lead poisoning can affect which genes work. Pollution. You could have talked about identical twins and how that they get more and more different as they age. Stress. There's all kinds of things you could talk about, but most of you probably would have chosen to talk about the methyl and acetyl group options, which are up here in bullets, are essentially 1, 2, and 3. All right, I think that's where we're going to stop for today with gene regulation. Before we do, let me take a minute and answer any questions that you guys might have.
I have a question.
Sure.
So basically for parts C and D, like D was asking about the epigenetic inheritance and then C was asking about the environmental factors, but aren't they, like, the responses pretty similar?
They would be similar. I think they just want you to be specific in, like, in D, you need to talk about methyl and acetyl groups. In C, you're talking about specific mutagens like UV light, gamma rays. You just got to be specific about address the question is what I'm saying. So let me pull back up the question real quick. Just a second to get there. So notice C specifically is talking about environmental factors that increase mutation rates. So there it's specifically asking about outside the body kinds of things, mutagens. So there you've got to talk about UV light or radiation or something like that. Whereas in G, you're talking about things that you inherit. So you can't inherit UV light or gamma rays. These are things you've got to inherit from a parent. So you can inherit certain methyl groups. You can inherit certain acetyl groups. You can inherit certain functional gene patterns because of the diet of your mother. That's what they're looking for. Does that make sense?
Okay. Yes, thank you.
Other questions that I can answer?
So for part A, when it says like in our response, if we put like the repressor proteins inhibit transcription, would you expect us to like have to write out like what transcription is or like how that relates, or is that like it's fine to say that repressor proteins just inhibit transcription?
Yeah, all you would really have to say there, and you can see the rubric on the screen now. Repressor proteins inhibit transcription. That one sentence is all it would take for that part of the question.
Okay, thank you.
Other questions?